All right. Parasites in the news. I spelled it. I missed it. Oh man, all this crap. Parasites in the news. God, crazy ads. Wolves keep brainworms spreading deer away from moose populations in Minnesota. So, caught this, I don't know, a couple days ago. Read it, remembered it. We'll talk about it. So this was, at least in the Smithsonian magazine. So in this cycle, you have a nematode that infects the brain. And it infects the moose. And when it infects the moose, it causes weakness, lack of balance, withering, or you could say loss of muscle tone and stuff, and eventually death. They pick it up by consuming some of the larval stages that are out in the environment. Deer can get it, but deer don't usually show the same type of symptoms. So you could say that deer could be almost like a reservoir host for this group, for the large moose. These moose were up in Minnesota. Wolf populations were decimated. It allowed the expansion of deer. Deer and moose started overlapping in their habitats. Deer are carrying the worm, spreading it around. Moose take a hit because of the parasites. Now, with the wolves coming back in that state, what they're doing is keeping deer populations down and minimizing the overlap, which is a good thing for these moose. So it's just one of those things where a lot of times you think about food supply, you think about maybe predator-prey interactions as regulating populations. Here, it's more than likely a parasite that is having an effect. I did have to laugh as I read through this. They got the life cycle completely wrong because they said they consume the eggs and then the eggs hatch in the brain. How did the eggs get there? The eggs aren't moving, so it's actually a larval stage. And how they get there, it's potentially that they get into the spinal column and travel up the nerve cord. So a lot of interesting stuff. But yeah, we have that here. So check it out. Pretty cool. Parasites in the news. All right. We have some housekeeping. I did change due dates for the quizzes. So check out the new due dates, get them done. That includes the lab quiz that was due on Friday. I forgot to change it on Tuesday or on Thursday, so on Friday morning it was changed. It's probably why you received another email, because I think one of the quizzes was unavailable that time. But get it done. We have lab today. We have 2 presentations, right? 2 presentations. Oh, you're presenting too? You're presenting, right? No, Jocelyn? Are you? That's next week? Okay. You're this week. 2 presentations. All right, we'll start with those. And then we'll have more presentations next week. I said I was going to get a presentation up. Yeah, that didn't happen. My wife was working from home. It's hard to give a presentation and get it quiet when everyone's home. So didn't happen. What we'll probably do is utilize some lab to get caught back up on our presentations. So bring your notes. We'll introduce at least Schistosoma and hopefully go over life cycles and stuff. All right. But at least we'll continue Schistosoma because I think we'll get to it today. Any questions? And if we don't get through everything for our exam, we don't get through everything. It is what it is. All right, so this is where we left off. We did our Paragonymous life cycle. All right, Paragonymous westermani lives in the lungs. Cook your meat. That's about as much as I can say. All right, so humans can get it. They can get it. All right, and they get it by eating undercooked crab and crawfish and undercooked guinea pig. If you're down in South America. And the worm goes right to the lungs. It's where they go. Right? So the pathology is really tied to, or the symptoms that we exhibit is really tied to the inflammation. And that's going to be the inflammation induced by both the worm and the eggs, because once they get into the lungs, they're secreting these eggs, and the eggs are going to have to get their way out. All right. So primary symptoms is result of that infection in the lungs. This is our pulmonary infections. Once you get that granuloma, normally inflammatory reactions start to cease. But until you get to that point, your body is fighting it. It's fighting the worm. It's fighting the eggs. Right? Now, if you get this capsule, this granulomatous capsule, right, in cells of the immune system, they encapsulate it. If that were to break, then you have potential for an increased hypersensitivity reaction, and it's going to be pretty significant. Is that common? I don't think it's necessarily super common. Normally you catch this before it gets to that point. The eggs, however, really pose the major issues because they're stimulating inflammatory response, and you'll start to get the development of what are called pseudotubercles. So you'll get this, immune cell complex around that egg, and it's almost like a seed for additional expansion of fibrin, and it actually resembles tuberculosis. Tuberculosis is you have this increased fibrin, reduces flexibility of the lungs and so forth. This kind of simulates it. But it's not tuberculosis, it's Plagiorchiformes or Paragonymous. Symptoms associated with that: difficulty breathing. You can get a chronic cough. Probably the more telling symptom is the sputum that has blood or brownish streaks in it. So you've got the eggs in the lungs. It's getting moved out by granulomas. And as it breaches into the actual lung space, you're going to have like a drop of blood that compete with it. So it's going to be a little bit colored. The brownish streaks themselves are the eggs. So if you are hacking up in your sputum, you've got these lines of, these brownish lines in there. Ask yourself, did I have undercooked crab at any time? Right? It's a possibility. Probably not here. It's mostly Southeast Asia. All right, so that's pulmonary infection. Occasionally, the wandering juveniles will also cause some damage. So the wandering juveniles are when you get these metacercaria. They penetrate out of the gut, and they're trying to find their way to the lungs, but they're having difficulty doing that. They are directionally challenged, is what you can say. And they're going to go to different organs of the body, and whatever organ they go to, you're going to produce different symptoms. So if they happen to go into the spinal cord, there's a potential that you'll have paralysis. Now, maybe temporary paralysis until the worm leaves, or could become permanent. Goes to the heart. You can have inflammatory reaction, inflammation of the heart, myocarditis. If you go to the brain, you get symptoms of cysticercosis. We haven't talked about cysticercosis, but this is a tapeworm larval stage. It gets it into the brain. You can have headaches, development of what you would perceive as migraines. You could have your pressure buildup, so disorientation, issues with balance. A severe case would be coma and ultimately death. All right, so as we talked about, once our immune system encapsulates those worms, then normally we stop responding, or at least we're not getting a strong inflammation response. And that explains why clinical symptoms will last for about 5 to 6 years and then decline to where you don't see them anymore. But no, these worms are going to live 10 to 20 years. And that is a strategy. You live a long time, means you can get a lot of eggs out into the environment, which increases your chances that you can get into that host, into that snail host. All right. And I know we went over the life cycle and we talked about the Microcircus Cercaria. This is one of these. That's the tail. It's super small tail. Any questions? There it is. All right. Opisthorchiformes, new order. Members of this order have testes that are at or near the posterior end. Cirrus and the cirrus sac are usually absent. The metroterm and the ejaculatory duct end up merging to form a common genital duct, and that's kind of why, if you have cirrus and cirrus sac absent, you're going to see that. And again, this is general description. Notably, the eggs are embryonated when they leave the definitive host. So as soon as they get out into the environment, they are ready to infect. But those eggs are only going to hatch after they get ingested by a snail. So the eggs are ready to are ready to infect as soon as they come out, but they have to be ingested by a snail to infect that next host. The family that our worm is in is Opithorchiidae. The adults are in the biliary system of reptiles, birds, and mammals. What does that mean? Bile duct. Bile duct, gallbladder. Another one that's in the bile duct and gallbladder. There are three species in these two genera, Clonorchis and Opithorchiformes, that are responsible for infecting over 300 million people a year. You can call this somewhat of a common parasite, at least in humans. Now, there's a lot more genre that are out there that infect wild animals, but we are going to talk about Clonorchis. And its effect on humans. Ready? There it is. All right, so the example is Clonorchis sinensis, which is one of these that infects the humans. We got our diagram, morphology of the adult. We've looked at it in the lab, so hopefully you remember it. This is the Chinese liver fluke. That's its common name. So hopefully you kind of recognize that when we're in the liver or the gallbladder, bile duct, all right, they usually take on a common name as a liver fluke. We've got the giant liver fluke. We've got the Chinese liver fluke. We have the lancet liver fluke, right? The dicrocelium. This is widely distributed in Japan, Korea, China, Taiwan, Vietnam. Hopefully none of our Olympians contract this while they're there. My guess is no, because they're all like super healthy eaters. However, if they go for that raw route, definite possibility. All right. So we're going to find these adults in the liver, bile duct, and gallbladder. It's interesting that these guys produce up to 4,000 eggs per day for at least 6 months. Massive egg output. It's not the highest. We'll see some that really dump eggs, but they produce a lot. All right. So let's go over this. We will go through our life cycle. Mute that. I'll use another piece of chalk. All right, so our definitive host is humans. That's going to be our definitive host. Our eggs, we are basically in the bile duct, in the liver. But we're going to release the eggs out into the environment. So our egg already has a myricydia developed inside of it. Myricydia. It's already developed inside that egg. It is ready to infect. And it infects once the egg gets consumed by our first intermediate host, which is a snail. Quadrant snail. All right, the egg will hatch. Myricydium will then penetrate out of the gut into the digestive tissue, where it transforms into a sporocyst and then redia. Now, this is one where I've looked up to try to figure out, is it like a mother-daughter sporocyst, mother-daughter redia? There wasn't a huge amount of information on it, so know that our redia is the cercarial producing stage for this parasite. All right. These guys, redia, then release our cercaria that are in the water and swim around. They swim around until they find and penetrate the second intermediate host, which is a fish. Specifically a cyprinid fish. So fish we can break down into different classifications. Who said zoology? He's a fisherman. Cyprinids are what type of fish? Give me another type of fish. Give me an example of it.
Gar.
Gar?
Gar.
Is it cyprinid?
Throwing out a guess.
It is. It's a good guess. Pretty certain it's a no for gar.
Is it catfish?
Interesting. Good guess. It's not a catfish. What did you bigger fish feed on? Minnows. Cyprinids are, collectively various minnow species, of which carp are cyprinids. So, say minnow species. So they're going to penetrate these fish where they insist. We're going to look at that gar. That has me intrigued. So we're going to be encysted metacercaria. It will be in the tissue, so it'll either encysted underneath the scales or in the muscle. And then they play the waiting game until our lowly human so our lonely human comes around and says, I'm going to eat fancy tonight, I'm going to eat sushi, and they eat these metacercaria. Now, our metacercaria will exist in the duodenum, and then travel into the bile duct directly. And there are some reports where they will go into the liver. So bile duct and then penetrate and get into the liver. Here is where they mature. And it takes about 1 month before the eggs get released, before they mature enough to release the eggs. No guard non-cyprinids. All right. So it takes about an egg, or about a month. These guys can live up to 8 years. They can live up to 8 years, at which point they are just dumping out eggs. They're dumping out eggs. Now, this looks like it's humans. They are the primary host. All right, but there's a question of whether any fish-eating mammal can be a host. There's a question, does any fish-eating mammal, can they serve as a host? And the reason we say that is because it seems to suggest that dogs and cats might be important reservoir hosts. So for us, if we just say it's humans, that's good. We keep with it. They're the primary ones, but perhaps we've seen, through several hundred years, some evolution of where they can now survive in cats and dogs, and they are what's causing them to persist. All right, so adaptations for transmission. One, they live a long time, increases chance that our eggs are going to get out and get consumed by the snail. Number two, these metacercaria are actually pretty resistant. Pretty resistant. There's a reason why I always say cook your food. These metacercaria can survive salting, pickling, drying, and smoking. Good thing salmon are not cyprinids. You see them. Pretty resistant. They cannot survive cooking. So you think, hey, we're gonna dry this fish. That's gonna kill everything. We're gonna salt our fish. We're gonna pickle it. That's great preservative methods. I mean, they are great preservative methods at keeping off bacteria and yeast, but they don't do anything with parasites. So, great adaptation to survive all of that. I think probably the most interesting one is the adaptation in Cercaria. So the Cercaria need to get to the fish. What do we know about Cercaria? They have a finite energy source. All right? So their infection windows depend on how active they are. The more active, the shorter the window. The less active, the larger the window. What these guys do, they come out, here's their tail, they come out and what they will do is slowly fall through the water column until they reach the bottom. Once they reach the bottom, they will then swim back up closer to the top of the water column, where they will then proceed to sink back down. Now, the R factor is kind of a measure of resistance in the water, and it's going to be fairly high for small organisms. So their rate of fall is going to be really, really slowly, which means they're inactive. They're not expending a whole lot of energy until they swim back up to the top. And in this way, they're able to stay around long enough for a fish to swim by. Now, how do they know a fish swims by? Because at some point, these guys will be swimming back up to the top. There's their tail. They'll fall, and then they will detect a water current along their fall. And that will stimulate swimming again, start getting them to move. So if they don't sense a current, to their knowledge, there's no fish around. There's nothing there. They're just going to slowly sink, and then all of a sudden, if a fish comes around, or something, doesn't have to be a fish, comes around and produces just a little bit of current, they sense that, and they start swimming. The more you're swimming, the more likely it is, it's a fish, the more likely it is that as they swim around, they will contact that fish. And as soon as they contact that fish, they clamp on. They use their suckers, they hold on, and then they try to find their way between the scales and penetrate into the muscles. Pretty cool adaptation. That's not the only types of adaptations that we see, but it's an example of some of our cercaria. Hopefully some of the snails that we look at, the honor students look at this year, we'll see some of these where they hang in the water. They sit in the water, and then we can kind of drag a forceps, tip of our microforceps across the water, and you can see when the current hits, all of a sudden they take off. So that's kind of a strategy.
Are the snails terrestrial or
They are aquatic. Aquatic snails. All right. Questions? That is Clonorchis. Ooh, this. All right. Pathology. If you get it, disease name is Clonorchis. Again, clever. It's linked to inflammatory responses just like our other liver flukes. And just like our other liver flukes, you've got basically irritation of the lining of the bile duct. Can lead to gradual thickening of those ducts and eventual occlusion. All right, so we've seen this before in some of our other liver flukes. Same types of symptoms. Kind of key for us is kind of knowing how the transmission may have occurred. All right. Perforation into the liver parenchyma is also possible. So it could move out of the bile duct in the gallbladder, get into the liver, and start causing some holes. All right. A lot of that is atrophy and necrosis. So pressure will cause death of the cells and atrophy and so forth. And if you have that, you have some liver function that'll be diminished. You're not as good at detoxifying some of your blood and anything else that the liver is going to be doing. Occasionally, the eggs, they end up, instead of passing out of the host, they go up and get caught in the gallbladder, at which point they could be nuclei for the formation of gallstones. I was kind of mean the first time my wife was diagnosed with some gallstones. I said, "You might have Clonorchis." She wasn't amused, as she's trying to pass them. Don't do that. Whatever you do, don't say that. You can think it, don't say it. All right. This last part is probably the most important. So for some reason, cancer, gallbladder cancer, seems to be associated with infection. So if you have Clonorchis infection, it almost seems like you're more likely to develop this form of cancer. Why? Good question. I haven't looked it up in the last year or two to see if we've learned more about it, but if you're interested in it, feel free. Check it out. Any questions? Super. Yeah, I know sushi is usually not really freshwater fish, but same thing. You can pick up nematodes from sushi. Pretty nasty ones too. All right. No questions? All right, let's move on. So our last digenetic trematode to discuss is Schistosoma. So we're moving to another order Strigeiformes. And I have a presentation just for this. So I'll have to get this, these life cycles, I'll have to get the quiz up for the end of the diversity one. And I think what we'll do on Friday is have an in-class quiz, which is, I'm going to have you diagram a life cycle. So we can kind of practice, you can see how we're grading and so forth. And that's going to be a prequel to the exam. All right, so Strigeiformes, members of this group, many different families in this order, big order. Ooh, let me close that out. The superfamily we're going to talk about is Schistosomatidae. Basically, there's 3 families in this superfamily that constitutes the blood flukes. And this sets them apart from the other digenes that we've looked at. So all the other digenes have been basically in the GI tract. Yeah, bile duct, but it's attached to the GI tract. I guess you can also say Paragonymous was in the lungs, so yeah, I guess that's different. These are in the blood. Members of the Schistosomatidae are the blood flukes of the birds and mammals. The Spirocheidae are the blood flukes of the fishes and turtles. And the Sanguinacolidae are blood flukes of the fishes and turtles. I have the asterisks basically are the dominant ones. So the Spirocheids are dominantly in turtles. The Sanguinacolidae are dominantly in fishes. Members, these blood flukes, have no second intermediate host. So they're going into the snails, they're developing in the snails are releasing cercaria, and then the cercaria are going to be penetrating directly into that next host. It's also rather interesting that most species in this group are dioecious. What does that mean?
2 separate sexes.
2 separate sexes, right? All the other ones that we've talked about are all monoecious. These are dioecious. So on our slides, we have male, let's see here. We have female worms in our slide collection. So, why be dioecious? What's the advantage?
Diversity.
Diversity, explain that.
So, female and a male both combined to create a more genetically diverse organism.
Okay. And how do you increase diversity through sexual reproduction? In addition to that, recombination events,
Oh, yeah.
big time. Yeah, 2 different individuals, you get 2 different lines. Again, you can increase diversity, but it comes at a cost, and that is diversity. If you're in an area that you're really adapted for, you're going to be more likely to break up those positive genes in a dioecious system. So, is one better than the other? Not really. Evolutionarily, not really. All right. So we are at the Strigeiformes. That's carp, common carp, cyprinid. All right, so we're in the Schistosomatidae. All right, we're talking about Schistosoma. We're going to talk about Schistosoma because it's an important parasite of humans. It's a causative agent of diseases called schistosomiasis, bilharzia, Katayama fever. These are all, again, caused by Schistosoma. This is the second most impactful parasite in humans. Second most impactful. What's the first? Plasmodium, malaria. So this is a parasite that still harms us. There's 240 million infected worldwide. Another 700 million individuals are at risk, meaning that they are individuals that live in the endemic areas. And most of these are in tropical and subtropical regions. So if you go up to northern Alaska, you go to the poles, yeah, you're not going to encounter this. But you're in tropical, subtropical zones where there's water and snails, yeah, you could be at risk. Now there's three main species that are going to infect humans, and we'll talk about all three of them. Schistosoma mansoni, Schistosoma japonicum, and Schistosoma hematobium. Schistosoma mansoni is primarily Africa, Egypt, and South America. This is its distribution. We do have intercalatum spotted on here, but everything in black is mansoni. You can catch it up near the Nile River Delta. It's up there. Right? It's crossed the ocean. It's crossed the ocean. There's actually some rather interesting theories about how it did that. If you're interested, go look it up. Schistosoma japonicum is China, Japan, and Philippines. So I should say, we're going to talk about the life cycles. We're going to do, I think, mansoni is an example life cycle, and then we'll talk about these two as how their life cycles diverge, or actually is how they're different. That's the important part. All right, japonicum, China, Japan, Philippines. I'm going to say the Far East. So japonicums out in those areas. And then Schistosoma hematobium also infects Africa and really does infect the Nile River Valley. Mansoni has made it up there, but you see this overlap in where they occur. Now, there are other species that can infect humans, including Schistosoma mekongi, guineensis, and intercalatum, but we're not going to talk about them. We're just going to talk about mansoni, japonicum, and hematobium. Ready? All right, so these worms, the adults are long and thin, and the reason is that they're living in the blood vessels. That's where they live. They're the blood flukes. So you have to be long and thin to fit inside of those blood vessels. They're going to be about 10 to 15 millimeters in length, so 1 to 1.5 centimeters. They are dimorphic. They're dioecious. All right? They're going to be dimorphic. Males are going to be larger and thicker. Females are going to be much thinner in size. Thinner in size. All right, so the males are going to have a gynecophoric canal. And that's the reason for its name. So schisto is basically schism. That's what it's named for. And it's named for this canal because a female it's going to lay inside of that canal. And basically, they're going to mate for life. They're going to mate for life. Your males, I'm going to say they have 5 to 9 testes, no sears, no sear sac. We're not going to, that's the number of testes helps distinguish between the species. We're not going to worry about that. But females will lay inside of this gynecophoric canal. All right. And it's important. These worms in our life cycle, they have to pair up. The female has to get into this gynecophoric canal in order to not only mature, but also to find their way to the site, to the final site of infection. There's something about pairing up that makes these worms better able to move through the blood vessels. If they don't pair up, they don't mature. They don't mature into adults. And these worms live 5 to 30 years. They're long-lived. And again, good reason. They have to live long to increase the number of eggs that get out of the host, or give them time to get out of the host. We don't see any fairings. I don't think we can really even make them out in our specimens. So, I just kind of for completeness, we see some of these things. Ready? In 5 minutes, we will not get through this life cycle. So how about this? Why don't we do the life cycle in lab? Do the life cycle in lab. We talk about the variation in our species. And probably getting the eggs out so we can pick up some of our missed material. And in that way, we have slides of the eggs, and we have slides of the adults that you can look at. All right? No point in trying to start and then having to end before we even get halfway through the life cycle. This will be one of the longer life cycles that we have. Not the longest. We got more in-depth ones. All right. Lab, 2 o'clock. We'll pick up with the life cycle. We'll actually start with our presentation. So if you're presenting, make sure you email me your presentation so I could get it on the computer, and then I'll be able to post it. Get it posted after lab. What's that? Yeah, I responded.
Yeah, but I only have a little bit of questions.
Okay. Yep. And this.